Nassau Grouper are an icon of the Caribbean.
You see them in artwork, they're on postcards, they're on the covers of dive magazines. They're representative of the Caribbean.
For most of the year, Nassau Grouper are solitary creatures, each inhabiting its own territory on the reef. But during the winter full moons, the fish gather to spawn.
Nassau Grouper form absolutely massive spawning aggregations, historically in the tens to perhaps hundreds of thousands.
It's just really a natural phenomenon.
An age-old ritual. Their annual dance occurs at dusk. These days, spawning Nassau Grouper have become a rare sight.
Primarily because of fishing at spawning aggregations, the numbers of Nassau Grouper throughout the Caribbean have declined dramatically.
There's so few left that an aggregation of a couple thousand fish is about as large as it gets.
Any spawning aggregation should be protected because that's the only time of the year that fish actually reproduce.
Major funding for this program was provided by the Batchelor Foundation, encouraging people to preserve and protect America's underwater resources, and by Divers Direct, inspiring the pursuit of tropical adventure scuba diving.
Part of the British West Indies, the Cayman Islands are located in the western Caribbean Sea. South of Cuba and northwest of Jamaica, the island nation is made up of Grand Cayman, Little Cayman, and Cayman Brac. Tourists come from all over the world to dive on the spectacular walls and see the diverse reef fish, including the often very tame and friendly Nassau Grouper.
Divers love fish that are basically like big puppy dogs. They're not frightened of divers, they follow them around.
They come up to you and want to rub on you or have you rub them, and they'll just hang with you an entire dive if you let them. So they're just friendly.
The Nassau Grouper represent to a lot of people what a healthy reef should look like.
Groupers are top-level predators, and top-level predators in any ecosystem help to control the structure of the ecosystem.
And when you take out the apex predators, everything below them on the food chain gets disrupted.
Nassau Grouper are found in Bermuda, Florida, and throughout the Bahamas and Caribbean Sea. It is estimated that their population has declined by at least 60% over the last 3 decades.
It's a species that's been caught by fishermen for many years, so you see it more on plates than you see it in the water.
The International Union for Conservation of Nature now lists Nassau Grouper as an endangered species.
We have reached that threshold. We've knocked enough of these populations out that we're now on life support. Every single population that's reasonably large is absolutely critical to whether or not this species exists into the future.
Targeting Nassau Grouper at their annual spawning aggregations makes for an easy catch, but this has had devastating impacts on the population.
There were dozens of aggregations throughout the Caribbean that have been documented that were then subsequently fished to extinction, basically.
In places like the Bahamas, for example, they had something like 23 historical aggregations, and they have about 5 viable aggregations now. Cuba had about 22, and they have anywhere from 3 to none left. Places like Bermuda that had spawning aggregations, their aggregations have never recovered.
In the Cayman Islands, the Department of Environment has been studying Nassau Grouper since 1986.
The initial research helped us to understand that there were declining trends all around, actual catch numbers as well as in the size of the fish.
Historically, 5 spawning sites were known in the islands, but most had been fished to exhaustion. Then, in 2001, fishermen rediscovered an aggregation site or grouper hole on the west end of Little Cayman.
This is the last stand. This is the last large aggregation that we know about of this species in the Caribbean. In that first year that the aggregation here on Little Cayman was found, fishermen over the course of about 10 days removed 2,000 fish. And the following year, the fishermen came back and removed 2,000 more fish. So the original estimates were probably that aggregation had about 7,000 fish on it. And over those 2 short years, they took out 4,000. And that year, the market got glutted. You know, there's no place for 2,000 Nassau Grouper to be used in a small island country like the Cayman Islands.
A lot of it's spoiled, and it's basically the case where they just caught so many fish they couldn't sell it. They're catching it because the fish were biting, not because the demand was there.
The Cayman Islands Department of Environment, or DOE, was monitoring the fishing on the aggregation. And by 2003, a temporary 8-year ban was established to protect all spawning sites in the Cayman Islands from fishing pressures. That ban was renewed for another 8 years in 2011.
It takes 8 years as kind of the average for Nassau Grouper to become reproductively active. And so they thought they'll give them 1 generation to be able to figure out, are those protections what's needed?
To answer this and other questions, the DOE teamed up with experts from the U.S.-based Reef Environmental Education Foundation. Reef conducts research with the help of volunteer scientists and divers. Together, the DOE and Reef formed the Grouper Moon Project in 2002. From the offset, the scientists wanted to know how many grouper attend the annual aggregation and if closing the fishery leads to a recovery of the species over time.
When you have thousands and thousands of reef fish on a deep reef, and you're trying to count the number of fish that are there, it's quite challenging to do. We tackle that problem in a number of different ways. We tag fish on site with small floy tags that are basically little streamers.
So it's a small spear that we use and shoot the side of the fish just below the dorsal fin and inject the tag just deep enough to get it to stay without bothering the fish too much. Those fish that we tag move around enough within the spawning aggregation and mix enough with all the other fish. They're distributed randomly among all the other individuals. So we tag a certain proportion of the population. We don't know what proportion that is. We just put out 100 tags. Theoretically, then afterwards, if we count sets of 50 fish and we see 1 tag on average, and we do many, many counts like that, and it comes out 1 tag in every 50 fish, we know that 100 tags would probably represent 1/50th of the total population. So we can multiply 100 tags times 50 and get the entire population. That'd be 5,000 fish.
Scientists also count fish in videos they record at the site. One year, they even used sonar to estimate fish numbers.
And lo and behold, the numbers came out pretty much the same.
Another way to determine whether the population of Nassau Grouper is recovering is by looking at the sizes of fish at the aggregation site.
So we've been going out there every year since 2004 with a special video system that has a pair of lasers on it that are set 25 centimeters apart and they're perfectly parallel. I put the lasers on the side of the fish, and then all of a sudden I have a scale on the fish that I can use to measure.
Using computer software, scientists can determine the length of the fish based on the known distance between the 2 laser points.
We go out and we try to measure several hundred fish every year and look at changes in length distribution. And what we've seen with that is that in 2004, 2005, and 2006, length distributions were shifting towards larger fish. So the fish were growing in size, but no new young fish were coming in. And then in 2007 and 2008, that length distribution started shifting, and we know, based on the measurements we did, that we still have the same number of big fish, so therefore we must have a lot more small fish. If we have a lot more small fish, the population is increasing in size. And so we're using that as one way to look at whether or not we're getting more fish into the population.
Another key question the scientists needed to answer was where the fish at the aggregation site were coming from.
There was lots of theories by the fishermen that the fish that showed up on the spawning sites weren't all from Cayman. They came from other countries, they came from Jamaica, they came from Cuba, and so the only time that we had to fish them was when they were at the spawning sites.
To test this theory, experts put hydrophones around the perimeter of Little Cayman that could receive signals from tagged fish.
We basically got acoustic transmitters and we surgically implanted them into the stomach cavity of the fish. What we wanted to do was we wanted to find out the fish that we tagged at the site, are they going back to home reefs around Little Cayman, or are they actually leaving and going someplace else? So it turns out that what we were finding was all the fish that were tagged are in fact resident around Little Cayman only. None of the fish actually left.
If they're only coming from this island, it means that that aggregation is critical to the local population itself, and it's a time when those fish are most vulnerable.
And that's really important information to have when you're trying to convince politicians and decision makers to make decisions that are going to be not universally popular.
It is still a mystery how the grouper know when to move to the spawning site.
They usually will show up in January, February, and sometimes in March. Most years, almost all, if not all of the spawning will only happen in January or February.
While at the aggregation site, the Nassaus display a variety of color phases.
The Nassau Grouper regular coloration is that brown and white barred pattern with the big saddle at the back.
Then there's white-bellied, and that's typically taken on by Nassau Grouper both on their home sites and out there during spawning when they're over a white area or they're up in the water column. It's basically a backlight camouflage. And then there's black and bicolor, and those are the two colors that fish are almost exclusively in when they're releasing gametes.
Spawning occurs between 1 to 8 days after the full moon.
At this point in time, it really is a mystery. We do not know what it is that they are sensing, but clearly they're smart enough to sense something because they spawn on particular nights.
One theory was that spawning may be influenced by the currents. To test that idea, the scientists used satellite drifters to track the movement of the water.
Which is basically a big sock that hangs down in the water, and it's tethered to a cable with a big ball on the surface. And in that big ball, there's a satellite phone and there's a GPS. And every 10 minutes, that satellite phone just sends us a text message and says, "Here I am." By deploying those on the aggregation when the fish spawn, we could see what the potential path is for the babies as they're leaving the aggregation. And we did this for 3 years on nights that they are spawning and nights that they aren't spawning. And what we found was that on the nights that the fish aren't spawning, the trajectories kind of go off to the north or to the west or sometimes to the south. And the nights after spawning, they kind of go off to the west or to the north or to the south. But on the nights that the fish spawn, the current patterns change on the aggregation site so that the fish are spawning exclusively, as best we can tell, on nights where the ocean currents are working to keep their babies close to home, which is right here on Little Cayman Island.
The fish larvae typically drift around the open ocean for 35 to 40 days.
And after that, the juveniles tried to find suitable habitat. And some people think that they can actually hear the sounds of waves crashing on reefs and things like that, and they can orient towards that so they know which direction to swim. And then they'll come in over the fringing reef, and they'll look for small areas where they can hide and grow. And that's sort of the two things they need to do, is eat and not be eaten. And so for their first year or two of life, they may be in these really shallow areas, and then as they grow, they tend to move offshore into deeper habitats until they become the adults, find their way out to the aggregation, and repeat the cycle all over again.
I think based on what we saw earlier today and the fish, the behavior, the way they were acting, it's going to happen. Go Grouper!
There are definitely a lot of courtship behaviors.
The large females that are very hydrated with eggs, you'll actually have a group of males that will follow her around and nudge her and try and encourage her to go up and release her eggs. So we actually see that herding or shadowing behavior. And it's almost like they're harassing her into going up. And finally she said, okay, I'll go and do it. And then she, that's when she'll shoot up, and then those males will follow her.
You'll see spawning start to happen about 10 minutes before sunset, and then it really starts going gangbusters right at sunset. It starts to become a melee. So you'll get a reasonably large individual releasing gametes, and you may have as many as 40 or 50 bicolors following at the same time and releasing this giant burst of gametes as they move up into the water column. When you start the dive, your visibility is over 100 feet. Towards the end of the dive, it's more like 20, and it's because you're swimming in a sea of eggs. It'll push on until about 20-30 minutes after sunset. And right at the point where it's truly dark, all the fish will just drop down onto the reef and find a hole to hunker down at night. They'll usually spawn for 3 nights, and then they'll go home. And it'll literally go from several thousand fish to no fish.
They were there. They were rocking and rolling. Very cool. Very, very cool.
There were just spawning bursts everywhere you looked. It was amazing.
Several of the divers collected eggs released in the spawning burst with a plankton net.
And if you look very carefully in there and there and there, you can see those little pearlescent drops, and those are eggs. And the cup's full up to there with eggs that we just collected off the spawning aggregation. We're hoping to be able to go through and do a genetic study on these eggs and figure out how many different females are out here spawning. We can then come back and look at all our juveniles that are around the island and see whether or not the juveniles are related to the adults out here.
For several years, the scientists had searched the shallow areas for juvenile Nassau Grouper without success. Then, in 2012, reports came in that little grouper had been sighted close to shore.
So we said, let's try and catch some of the juveniles so we can get genetics from it, and we can see whether they're related at all to the fish that are spawning out there, and maybe close that loop to say, you protect the fish out there. That will then result in fish coming back to your own reefs.
To locate the small young grouper, snorkelers cover a wide swath of shallow water. Once they see a fish, the GPS location of its hiding hole is recorded.
Okay.
Next is the rather difficult task of trying to catch the little grouper. Once a grouper loses the chase, it's brought back to the boat to be measured and have a fin clip taken.
And what that does is allow us to analyze the DNA.
Genetic data collected previously from across the Nassau Grouper's range indicates that fertilized grouper eggs can travel long distances.
Preliminary results are suggesting that there's a lot of genetic exchange between populations. So even with the work that's being done on drifters showing that, there's a potential for larvae to be retained close to shore, there's actually a lot that are being taken in currents to other regions in the Caribbean.
The question of whether or not there are larvae coming from other places to here is a tough one to answer. The only real link that's been shown for the Cayman Islands is between here and the southeast tip of Cuba. We don't think that there would be a lot of import of Nassau Grouper into this area. Upstream of here in Jamaica, and word out of Cuba is that there's just not enough Nassau Grouper left there to expect decent amounts of larval recruitment here from those locations.
Given the low chances that grouper larvae from other islands could make it to Little Cayman, experts feel it is all the more important to protect the local fish, with hopes that they will replenish and sustain the population.
In the 8 years since we've closed the Little Cayman site, we've seen an increase of about 1,500 fish. That's not a lot of fish.
On average, they reproductively become active somewhere between 5 and 7 years old, and they'll live anywhere from 20 to maybe as much as 30 years. And because it takes quite a while to get to that reproductively active stage, recovery, once conservation efforts are put in place, can take quite a while.
The research is also suggesting that larval recruitment, or the number of larval fish that will survive and settle in shallow water, is very sporadic.
We can go many years where there's no recruitment or very little recruitment, and then you may get 1 year where there's literally hundreds of Nassau Grouper that show up in the inshore seagrass beds and nearshore reefs.
Knowing this, scientists are suggesting the implementation of more stringent, long-term fishing regulations.
The Department of Environment has been using the science to come up with a management recommendation that we feel will work for the long term. And what we believe we need to do is establish a closed season during the spawning period, so where fish cannot be taken anywhere within our territorial waters. So that closed season would run from November through the end of March. And that's what we've proposed to government.
The DOE is also recommending that all the known spawning sites around the Cayman Islands be closed to any fishing year-round.
We've seen a number of species aggregating there to spawn. So we think that they're important areas generally for fisheries around the island. The other thing that we're trying to do is to give a realistic, sustainable catch limit. So we're thinking that it would be 1 to 2 fish per person per day, which is a low number if you're used to catching 60, 70 fish a day on the aggregation, but we believe that's what we have to do.
We're not about stopping fishing, we're about ensuring that this can go on for generation to generation.
While there is no commercial fishery for Nassau Grouper in the Cayman Islands, there has been some opposition to the proposed legislation by artisanal fishermen.
In East End, they would salt the grouper and eat it very traditionally at Christmas time.
That's one of the things that a lot of the fishermen talk about is losing their tradition and their heritage.
But the majority of people now definitely feel we need to have these sites closed.
On these islands, nobody's starving. They don't have to depend on the grouper to feed their family. There's plenty of jobs, and if a man wants to make a living, he does not have to make it from catching a grouper.
Caymanian native Bruce Eldemeyer has seen firsthand what intense fishing pressures can do to fish populations.
I was a commercial fisherman in the state of Louisiana for 26 years, and we wiped out the redfish and the trout over there. And I know what it's all about. You've got to have restrictions and laws to protect something like this.
The time now is to step up that protection to be able to protect them, into perpetuity.
To create greater awareness of the issues surrounding Nassau Grouper, the DOE and Reef have reached out to local schools to share their research findings.
We're doing lesson plans that focus on the ecological importance, but also the cultural heritage of the Nassau Grouper, and being able to make that connection so that they can see why it's such a special thing to protect.
Living in the Caribbean, it's very important for these students to be aware of the issues that are going on around them.
I mean, I knew there was grouper holes, but I didn't really realize how important the spawning was and how catching that much of them could lead to a depletion of their stocks.
In 2012, the scientists started live video web chats from the spawning aggregation with students in classrooms on Grand Cayman.
The students get to ask real scientists that are out in the field about what they're doing and how they're collecting their information and their data.
Here we are, coming up on the Nassau Grouper. There's a whole heap of them out here, as you can see. And these fish are ready to spawn. Got some the big friendly female.
Bryce, the question is, how many eggs does one female have?
It's certainly thousands and thousands of eggs. The bigger the fish is, the more eggs that fish produces.
Whew! It's a lot of fish down there.
Can't believe that we were able to share that amazing experience with everybody across the web and in the classroom.
The future of Nassau Grouper in the Cayman Islands looks hopeful. Scientists have worked tirelessly to better understand the lives of these amazing fish. Now they hope that their scientific findings will lead to permanent protections for this endangered species.
For many generations, our forefathers have eaten grouper as a cultural tradition, and so to continue that tradition, we want to be able to protect them so our children can see what groupers are, can take part in that heritage as well.
Major funding for this program was provided by the Batchelor Foundation, encouraging people to preserve and protect America's underwater resources, and by Divers Direct, inspiring the pursuit of tropical adventure scuba diving.